Thank you everybody for joining us. Today we are going to be meeting the UCSF Alzheimer's Disease Center team. Just to remind everybody, the Meet the Scientists webinar series is an opportunity to meet teams or investigators that are working to end Alzheimer's disease and other dementias. And it's also an opportunity for everybody, mostly for junior investigators, to learn about the exciting research that is happening with these teams, and also to inquire and learn more about potential career opportunities. So we are doing this monthly. So if you want to see more of the scheduling, you can visit this website. And just to remind you about the next two meetings. So we have in April, we are going to be meeting the Rush Alzheimer's Disease Center in April 7th. And in May, we are meeting the University of Kansas Alzheimer's Disease Center as well. So today is my pleasure to invite and host everybody from the UCSF team, as I was mentioning just right now. I'm going to start by introducing Dr. Gil Rabinovici. He's the professor of neurology at the UCSF Institute for Neuroscience, and he's also the lead and the director of the Memory and Aging Center, PET team over there. So I'm going to pass it to Gil, and thank you again, everybody, for joining us, and thank you to the team of UCSF for accepting this invitation.
Thank you so much, Akil. Really a pleasure to be here. Let me try to share my screen. See it okay? You see my screen?
Yes.
Yep, you're good.
Okay, great. So really a pleasure to be here. Oh, screen is stuck. There we go. So we'll be presenting our Alzheimer's Disease Research Center, which is embedded in the UCSF Memory and Aging Center. UCSF is the University of California, San Francisco, for those of us who are joining us internationally. The Memory and Aging Center is a division in our Department of Neurology. It is, in my very unbiased opinion, a very stimulating and a really fun place to work. Our director, you can see his face down there, is Dr. Bruce Miller, who founded the center in 1998. And really our mission of the center is to provide the highest quality of care for individuals with cognitive problems, conduct research on causes and cures for degenerative brain diseases, and educate health professionals, patients, and their families. And I think the mission of the Alzheimer's Disease Research Center is really aligned with all three of these values. So for those who aren't familiar, I know you've had other ADRCs present, but the ADRC consists of several cores. So there's a clinical core, a data management core, neuropathology, outreach and education. We have an imaging core, a biomarker core, and then a research and education component. And really the administrative core at the center's role is to integrate these cores into one center, and also to integrate them into the local ecosystem. So the community, other partners, and other research organizations. So our center was founded by Dr. Miller in 2004. One of our main goals is to support a lot of additional research. So we're currently supporting both primary and training NIH grants, as well as many non-NIH projects. The characteristic of our center is deep phenotyping of large and diverse cohorts. And in particular, we have an interest in young onset dementia, frontotemporal dementia, and early onset Alzheimer's disease. We also have very active cohorts in corticobasal syndrome and progressive supranuclear palsy, which are part of the FTD spectrum. And Stan Prusner is at UCSF, so we also have a very active prion and Creutzfeldt-Jakob disease program led by Dr. Michael Geschwind. Our primary outreach activities are into the Chinese and Latinx communities in the San Francisco Bay Area. You'll hear a little bit about that today. We're interested in not only cognition, but very interested in deeply phenotyping behavior, emotion, and social cognition. I think these are all critical across dementias, but are particular in frontotemporal dementia, and you'll hear a little bit about that. We obtain all the imaging, genomic, biofluid markers, as well as some cell lines, and we have a very active autopsy program for these different dementias. Even though this is primarily a clinical endeavor, we work very closely with our basic scientists, including Leonard Mucke, who leads the Gladstone Institute affiliated with UCSF, and Stan Prusner, who leads our Institute for Neurodegenerative Disease, And then we work closely with consortia that are interested in tau and frontotemporal dementia, work very closely with the Alzheimer's Association. And then we also have a variety of different training programs for clinicians as well as neuroscientists. These are the leaders of our course. So Dr. Miller and I co-direct the administrative core. Joel Kramer and Kate Possin lead our clinical core. Kate Rankin and John Newhouse, our data core. Bill Seeley and Leah Grinberg, our neuropathology core. I think you'll hear from both of our outreach and education leaders today, Serggio Lanata and Boon Lead Tee, sorry. Howie Rosen and I co-lead the imaging core. Amy Gao and Jenn Yokoyama lead our biomarker core, and then Christine Yaffe and Howie lead our research and education component. So we are part of the National NIA-funded Alzheimer's Disease Research Center program. There are 31 centers, I think, at the moment. Here you can see the distribution, and there are also 3 P20 grants now for centers that are on their way up to become full ADRCs. So hopefully we will improve some of the geographic coverage that we have in the United States. You can see that we have 3 centers in Northern California, but the identity of each center is really quite distinct. We're very collaborative. We work closely with a number of centers across the US, and I just wanted to highlight a few key programs. One is the ALL-FTD, Longitudinal Frontotemporal Lobar Degeneration Network. This is co-led by Brad Beauvais at Mayo Clinic and Adam Boxer and Howie Rosen here in the UCSF ADRC. I'm one of the co-PIs of the LEADS, Longitudinal Early Onset Alzheimer's Disease Study, that's led by Liana Postolova at Indiana, as well as Brad Dickerson at MGH and Maria Correa of the Alzheimer's Association. By virtue of my research, we work closely with the IDEAS program, which is a national study partnership with Medicare in the United States to study the clinical utility of amyloid PET. Kate Possin leads a very vibrant care ecosystem program looking at different care model deliveries. And then we have, again, a very active training program in behavioral neurology, neuropsychology, neuroscience, and are affiliated with a number of other grants. We also work closely with a number of institutions around the world, close collaborations with several centers in Europe. I particularly wanted to highlight this collaboration, which is a new grant called Research Dementia Latin America, or REDLAT, led by Agustin Ibanez, and Bruce Miller and Jenn Yokoyama might say a few words about this very exciting South American dementia consortium. And then a real game changer for us was the founding of a Global Brain Health Institute in collaboration with Trinity College in Dublin, Ireland. This is an institute that is dedicated to training the future leaders of brain health around the world. You can see we've had very diverse fellows from across 42 countries and also diverse disciplines. So not just traditional biomedical or cognitive neuroscience backgrounds, but also artists, social scientists, health economists, public health experts, and even folks from business and technology. We have a long tradition of collaboration with artists. I think this dates back to Dr. Miller's early career when he described some of the emergence of new artistic talent in the setting of neurodegenerative disease, frontotemporal dementia, and progressive aphasias. And this has really remained a very dominant theme in our center with a whole variety of different artists. You'll hear today from Josh Kornbluth, who's a very talented performer and filmmaker, about his Citizen Brain series. And then finally, in this very tumultuous year, I'm very proud that our center has stood up for some social justice issues, including opinion pieces shown here on homelessness, race-based norms and cognitive testing, science denial and conspiracy theories, and a very powerful video produced by our Black Leadership Advisory Council on the experience of being Black in the Bay Area and in the Memory and Aging Center. And maybe we can share some of those links with the people who registered for this webinar. So I'll end there. Thank you very much for joining us. And we have a really terrific lineup of scientists at the ADRC to briefly share their work with you today. Thanks a lot.
Great, Gil. Do you want to call the next person?
Can people see my screen?
Yes.
Yes.
Excellent. Hi, I'm Harli. I'm the program manager for our Alzheimer's Disease Research Center. And as you can maybe get from Gil's presentation, we do a lot of different things. And so we invited various different postdocs and junior and senior scientists to give very brief snippets of what they research to give you a sense of all the different directions we look at brain health. And so with that, I'll be driving as the manager, which speaks to my role, and people will give you a brief intro.
1 minute, Harli, is that right?
Yep, 1 to 3 minutes. We're referring to it as tapas instead of a full course.
Okay, fantastic. All right, well, I'm Kate Possin. My work is focused on improving diagnosis and care for Alzheimer's and related disorders. Central to the diagnosis work is a software platform that we've homegrown here at UCSF called TabCat. It's a platform for administering cognitive tests and other clinical tools. And these tools are useful in research with diverse populations and also in clinical settings. For example, we use them in primary care to help primary care providers to more accurately diagnose MCI and dementia. Currently, some of our work is focused on looking at the relationship of performance on this test to Alzheimer's biomarkers. So we hope they'll be useful for determining what is the cause of the neurodegenerative disease. And all these tests are very quick and on iPads. And then my other major area of research is the care ecosystem you just heard about. So this is supportive care for families living with dementia. And we test it through research, and we're doing implementation science work and policy work to try to make this a new standard of care across the country. And in general, I'm very much interested in designing and testing and implementing new care models to improve diagnosis and post-diagnostic care. I'm looking for fellows from all kinds of backgrounds. If you want to develop new care models or do neuropsychology work, you might have a background in nursing, social work, psychology, geriatrics, neurology, so pharmacy, but we're all kind of gathered together around these common missions. Thank you.
And
I'm next. Hi, I just wanted to give a shout out to Ucsf. Ucsf, thank you so much for inviting us. It's so good to see you. Ucsf and I know each other from our times in Boston, so it's just great to see the worlds connect. And my name is Elena. I am a neuropsychology postdoc. I work with Kate, primarily. Kate is my mentor. And my interests kind of all revolve around the issues of diversity and disparities in dementia. And I specifically am interested in the role of social determinants of health as potential mediators or moderators of racial disparities that we see. I just submitted a K-99 application, so I'm very excited, and it's specifically on this topic. But I just wanted to share a couple of highlights from our previous work. For example, in the paper that was published in JAMA Neurology just today, we looked at racial and ethnic disparities in dementia diagnosis, and we indeed found significant racial disparities, particularly in timeliness of diagnosis, meaning that racial and ethnic minorities were diagnosed at a later stage of dementia. And we also found substantial disparities in terms of how comprehensive was the actual workup, how many tests did people get for dementia, which is something that I think we all see in our kind of daily clinical care, unfortunately. And then another big part of what I do is looking at multicultural neuropsychology. I'm a bilingual neuropsychological provider myself, so I think a lot about development of measures that are culturally sensitive, as well as how do these measures relate to, as Kate mentioned, Alzheimer's disease biomarkers in particular. So we do a lot of work trying to understand what is the role of cognition and biomarkers in early diagnosis, and how can these developments, for example, in blood-based biomarkers in these brief cognitive tests, be used to facilitate early diagnosis across racial ethnic groups in order to, again, reduce those disparities that we see. And finally, one last thing, this map that I have on my slide is actually a very exciting collaboration that we have with the Global Brain Health Institute, where we collaborate with fellows from all over the world. And we just recently did the study that is under review now, where we look at how our brief cognitive measures, for example, such as MMSE, are used throughout the world by dementia specialists. How helpful are they, and what are the things that need to be improved? So this is just sort of a snapshot of what I do, but this is my Twitter handle. This is my email. Please feel free to reach out. Would love to connect.
Boon Lead, are you muted?
Thank you. Hi, I'm Boon Lead Tee. I'm an assistant professor at UCSF Memory Aging Center. I also help co-lead the UCSF Chinese outreach team with Dr. Serggio Lanata. So as what Elena has said, our center focuses a lot on research diversity. And one thing that we do recognize is language do play a critical role in cognitive assessments. To better reflect that, our group actually has adapted our cognitive battery to be more culturally sensitive as well as linguistically tailored for our Chinese cohort. For instance, on the right, we have this culturally relevant stimuli like abacus or chopsticks for Chinese participants, but we also have linguistically tailored tests like using lexical tones or logographic reading and writing tests for Chinese language users. And also in recognition of the rapidly growing multilingual population worldwide, as well as here in California, we introduced language questionnaires that help us quantitatively measure the linguistic background of our multilingual participants, so we can recommend optimal testing language that best reflect their cognitive ability, but also better phenotype their multilingualism characteristics. And on the left is our CLAP project. So thanks to the support from Alzheimer's Association, GBHI, and NIA, we are able to partner with multiple university and hospital at Hong Kong and Taiwan to study Chinese-speaking primary progressive aphasia individuals. And so through the project, we were able to identify numerous linguistic features that's critical for the diagnosis of PPA in the Chinese-speaking population. So for instance, we noticed that Chinese language user actually presents with different dysgraphia phenotypes when compared with English language users, which is important for classifying the different PPA variants. We also found that the strategy to evaluate motor speech performance in the Chinese-speaking population is actually through lexical tone, which is a linguistic features relatively absent in most Indo-European languages. We also aim to anatomically validate a lot of the multiple cognitive tests in our Chinese cohort, and hopefully our clinical evaluation can be more and more population specific. So shown here in the light blue on top is actually my email. We are also very interested in building cross-linguistic cohorts to promote language diversity in the field of dementia research. So please feel free to contact me for any question and discussion. Thank you.
And Serggio, we have you, but you might be muted.
Yeah, I'm here. Well, thanks for this opportunity. Just briefly, I want to tell you about this project. We're very interested in studying how different social and environmental factors lead to different brain health outcomes, including Alzheimer's disease, cerebrovascular disease. This project stems from the realization that in San Francisco, there is a lot of variations in social vulnerability across different neighborhoods of the city. This is true of the Bay Area at large too. And people that inhabit these neighborhoods, therefore, are, we expect them to have different health outcomes, including brain health outcomes. This map, we correlated with a map that shows where our current research participants into our ADRC are coming from. As you can see, the areas that are most vulnerable are the least represented in our ADRC. So part of this project is aimed at recruiting people or engaging people from these vulnerable neighborhoods. And that will allow us to study how embodiment of these vulnerability factors influences brain health. Next slide. And the overarching idea is that these brain health outcomes are not only influenced by these known risk factors, medical risk factors and their associated behaviors, but that these things don't occur in a vacuum. They occur in the setting of a long history of exposures to different social and environmental factors. So as a first step to this project, we finalized the creation of a social determinants of brain health questionnaire that is gonna hopefully lead to what I'm calling a vulnerability index at a participant level. We have the first iteration of this questionnaire in Spanish and English, And we've completed phase validity testing on these 2 languages, and we're hoping to start collect this data via Qualtrics and be able to draw these correlations. And the next step would be to have it done in Chinese as well. I think that's all I'm going to say. Thank you.
And Kaitlin, I'll pass it over to you.
Thanks, Harli. And thank you first to Akil and Renaud for setting this up. This is really fun to be a part of this. So I'm Kaitlin Casaletto. I'm a neuropsychologist at the UCSF Memory and Aging Center. And my lab is particularly interested in deeply characterizing individuals that show abnormally successful clinical outcomes in the context of aging and neurodegeneration. The general thought is if we can identify some of these protective factors, these could be potential therapy targets for cognitive aging more broadly. Harli, if you mind clicking through a little bit. Okay. And so some of the methods, the ways that we approach this is by case matching. So identifying people who show protected outcomes in the context of a risk factor like amyloid or white matter hyperintensities, and really trying to characterize them. I also focus really closely on lifestyle behaviors that we know are associated with successful outcomes. So like physical and cognitive activities. We use wearable data to try to capture some of these phenomenon. Another differentiator is a real focus on fluid biomarkers to capture different protein indicators of molecular biological pathways that we think could be differentiating some of these phenomenon. Of course, cognition as a neuropsychologist, and we're also very interested in sex as an important factor in this resilience phenomenon. And if you go to the next slide. So I just wanted to highlight some of the studies in the last year that I think are emblematic of how we're thinking about these different processes. One study in which we demonstrated that active lifestyles are associated with better clinical functioning, more cognitive resilience in individuals who carry an autosomal dominant mutation for frontotemporal dementia. We've also demonstrated a significant impact of sex on the relationship between physical activity and cognitive aging outcomes, including immune activation indicators. And then I wanted to highlight this last paper here on different blood factors. This is a translational research project that was occurred within UCSF itself, but we essentially were able to boil down the neurogenic benefits of exercise to a single molecule that's present in plasma and seem to have relevance in animals and in humans. So we're very interested in collaborating across sort of bench animal to bedside. Some ongoing projects here, if anyone's interested in getting involved in some data, the ACTAN study is the primary cohort in which we really deeply characterize lifestyle behaviors, wearables, plasma, cerebrospinal fluid. We also have an interventional study where we are manipulating these lifestyle behaviors and observing outcomes, particularly again on these molecular biofluids. And then a new direction that we're headed in is honing in on one specific biological pathway through synaptic functioning and trying to understand how synaptic functioning might be supported by behaviors like physical activity and then may in turn support cognitive resilience long-term. So if anyone's interested in wearable data, fluid biomarkers, lifestyle, cognitive resilience, please get in touch.
And Cutter, we'll pass it over to you.
Yeah, thanks, Harli. So my name's Cutter Lindbergh, and I'm a neuropsychology fellow at the Memory and Aging Center. And thanks to the organizers for inviting me to participate in this event and for you all for being here. I primarily work in Dr. Joel Kramer's lab here at UCSF. And my primary interests focused on sort of characterizing some of the earliest changes associated with age-related neurodegenerative conditions such as Alzheimer's disease, and trying to sort of predict who may be at risk for subsequent decline with the goal being of trying to identify folks who may profit from intervention, early intervention. So I'm just going to show a few studies here, 3 studies that we published over the past year related to this topic. So the first, you can see the image here on the screen, involved using a particularly sensitive memory test to attempt to predict cognitive and brain structural trajectories. And So all the individuals in this study were clinically normal at baseline. And the test that we used was special in the sense that most neuropsychological tests test recall for information after relatively short delays, like 10 to 30 minutes. But this test tests recall ability after a full week. So tapping into some of that longer term memory consolidation process. And what you can see is that performance on this sensitive memory test at baseline, even among clinically normal older adults, predicted subsequent cognitive trajectories and subsequent changes in medial temporal lobe volumes. We can click to the next. There we go. This was another study also looking in individuals who are clinically normal at baseline and looking at how amyloid burden as measured by PET imaging relates to cognitive trajectories and how this relationship is moderated by sex. So, and what we found is that females with elevated amyloid burden tend to be at greater risk for memory decline relative to males. And this was particularly true among female carriers of the ApoE4 allele. And we can go to the next. Perfect, thanks. The last study I just wanted to mention was using blood-based markers of inflammation, so TNF alpha, among clinically normal older adults. And what we found is that over time, older adults who showed increasing levels of inflammation in their blood, measured in the periphery, showed corresponding decreases in gray matter volumes and also increases in white matter hyperintensities and declines in cognitive functioning. So anyways, hopefully this gives you sort of a brief sampling of some of the work that we've been up to. Thanks.
Okay, and Renaud?
Hey, hi everyone. So I am a scientist. I work in Gil Rabinovici's lab. And so basically at the core of all our studies, we combine MRI and molecular imaging or PET to measure different types of brain pathologies and injury like amyloid and tau and brain metabolism. And a lot of our work consists in analyzing the relationships between these different measures and understand their spatial and temporal relationship with longitudinal studies. But then we also collaborate with basically almost every core and every group at the MAC. Notably, on the left, you can see the collaboration with the Neuropathology Core because we conduct a lot of PET-2 autopsy studies to really assess the sensitivity and the specificity of the brain imaging techniques to detect different pathological findings. And the whole goal is really to be better at interpreting the in vivo data that we acquire in patients. Then the bottom left panel illustrates our really deep interest for identifying the imaging underpinnings of the clinical heterogeneity of sporadic AD. And indeed, our Alzheimer's cohort is enriched in cases with so-called atypical AD. I don't think we really like this term, but so basically patients with non-amnestic syndromes or patients who develop symptoms early on in their 50s, for example. More recently, we've collaborated with the biomarker core to assess the blood biomarkers for Alzheimer's pathology. And we showed like in the on the top right panel, we showed really robust association between tau PET signal and phosphorylated tau measured in the plasma. So really with huge impact for more global and more inclusive research studies in the future. And last point I wanted to make is that our lab is really involved in various large-scale studies, including the IDEAS study. And so what's interesting is that the IDEAS study is meant to examine how amyloid PET impacts clinical diagnosis and care. But we also use this data for other more secondary purposes. And I just wanted to point this study, a very creative study by Leonardo Iaccarino, who's also in the lab. And he leveraged the fact that all these PET scans were acquired across hundreds of centers in the US. And he used that to look at the association between residential exposure to air pollution, measured based on zip code, and the rate of amyloid positivity. So as you can see, most of the studies we conduct in the lab are imaging centric, but really spanning a lot of different topics.
And Argentina, over to you.
Hi, so thank you for having me here today. And I mean, I'm very grateful to be at the MAC, and I'll say in a minute why. My background is as an electrophysiologist. I did my PhD studying synaptic plasticity and single molecule tracking, how AMPA receptors move from one place to the next in the neuron. And after a few years doing that, the MAC gave me an opportunity to totally shift gears in my career and do something really more applied to people and seeing patients and participating in something that has a more direct impact in people's lives than what I was doing when I was working with mouse models. And that made me very happy for personal and professional reasons. And exactly what I do there, I am the genetics and biospecimen manager. So that means I am kind of the gatekeeper of the samples and the data associated with those samples. So everyone in this amazing team, they see a lot of patients in clinical and observational trials, and they collect biospecimens. Then my team processes those, and we run a biobank, which is like a big inventory for these samples. And we also bank the data associated with those samples. Then we get requests for data and biospecimens from all over the world, really. So it's a very collaborative environment. And then we share those specimens and that data to make it available to other scientists that want to address the same questions on how to find better diagnostic tools and treatments for diseases like dementia.
Panos, over to you.
Okay. Hi there. My name is Panos Theofilas, and I'm a basic scientist and faculty at the Memory and Aging Center. My lab focuses on cell death mechanisms and tau pathological changes in Alzheimer's disease and other tauopathies. And to this end, I use a combination of iPSC-derived neurons from patients with MAPT mutations and validate disease markers involved in programmed cell death or apoptosis, such as caspases, in human postmortem brains from AD and FTLD patients. So what I did here is selected a few figures from 2 manuscripts currently under review as a summary of what I'm working on. And I'm happy, of course, to share the links to bioRxiv if you'd like to learn more. So going back to the slide, what you see in the top panel is iPSCs with the FTLD causing mutation V337M. And the grayscale images show the timeline of how an iPSC colony on the left is differentiated into neuronal precursor cells and then into mature neurons. Next, what you see is an immunofluorescent image panel that shows positivity of the mature neurons for upper and lower cortical markers, as well as the neuronal marker MAP2. So after culturing the cells for up to 3 months to imitate aging, what we see is that the mutant line shows significantly higher level of cell death compared to wild-type controls, overall suggesting an increased pathology due to the MAPT mutation. So in general, our data supports a model where tau pathological changes in the mutant cells activate caspases, which then cleave tau and promote cell death. So after these exciting findings, we asked if this pathway is also relevant to human tauopathies. So to answer this, Lea Greenberg's lab at UCSF provided human postmortem brain sections from patients with the same MAPT mutations as the cells I just showed you, and compare this to AD and control brains. So using multiplex imaging analysis with newly synthesized caspase cleave tau antibodies generated by Michelle Arkin's lab at UCSF, we found a positivity for all markers in both FTD and AD patients, but not in healthy controls as expected. So the results are summarized in the bottom right figure. So overall, our data suggests that caspase-mediated cleavage of tau could be an important therapeutic target against tau pathogenesis in tauopathies. And that's all I have for today. Thank you. And feel free to contact me if you have any questions.
And Jenn?
Hi, thanks everyone for the opportunity to tell you very briefly about our lab's research. And this actually addresses one of the Q&As at a high level, and you're welcome to reach out if you have more specific questions. So my work is mostly anchored in clinical and translational research in genetics and genomics. We do our studies both in the context of biomarker development as well as basic science discovery for new contributions to neurodegenerative diseases, with a primary focus on early onset Alzheimer's disease and frontotemporal dementia, although we also study late onset Alzheimer's disease and healthy cognitive aging. Our lab uses a number of different tools to do this, including imaging and genetics. In varying types of studies, we may look at gene expression from the brain, whether through public data sets or through our own primary generated data to look for relationships of gene expression in regions that are vulnerable or resilient to neurodegenerative diseases. We are also doing transcriptomics at a single cell level and bulk RNA sequencing in blood to look for gene expression signatures that correlate with disease. We are using genetic information from multiple different loci to ascertain multiple, polygenic risk contributions to disease, taking into account different variations and hopefully soon adapting these methods to more diverse populations. Of course, we use genomics for primary gene discovery. We have mapped some rare variants that contribute risk to disease. And one of the projects we're most excited about that has started recently is really building out both the infrastructure for research in other parts of the world, specifically focusing in Latin America, and also enhancing our genetic knowledge of these different populations, because we know that diverse populations are going to be needed to be analyzed in order to create discoveries that will be impactful for everyone. So if you have an interest in either collaborating or contributing or combining resources, especially for diverse populations, we're looking at Latin American populations, of course, also Asians as well. Thanks.
And to Jalayne.
Although we're going to take a little bit of a right turn now, but it really connects to the work that Dr. Yokoyama is doing. I am trained, I'm an assistant professor at the Memory and Aging Center and a trained lawyer and clinical ethicist, and I conduct empirical research on the legal and ethical issues across the Alzheimer's disease and related dementia spectrum. And my research really spans from the issues that arise anywhere from bench science all the way through what I deem translational plus, or sort of the issues that affect our patients and research participants as they try to take information about their diagnosis or risk into the real world and outside of the clinic. Some of the projects that my group is currently working on include looking at genetic data sharing and management and FTLD research. This is how do investigators share research data between themselves and between institutions, and what are the ethical issues that arise, including potential risks to participants' confidentiality. We're also looking at some of the issues related to return of research results, specific to research participants and ADRD. And we have a whole sort of some section of work focusing on questions related to the potential employment and insurance discrimination risks that might come from disclosing preclinical biomarker status to somebody who is asymptomatic. So how does that affect their insurance or their employment status? Working with transfers at UCSF, we're also starting to explore issues of payer coverage for things like ApoE genetic testing. And then we also have some work going on now that relates to dementia and the criminal justice system. Specifically, we're looking at how does the criminal justice system treat an individual who is either suffering from dementia at the time of arrest or develops dementia during their incarceration. And beginning to explore that area, which we are finding is much needed. I forgot to put my contact information on here, but I think, the only Jalayne Arias doing research in Alzheimer's. So feel free to Google me. I love working with students and people who like to think about these issues. We have a ton of data and a ton of things to be working on in this area. So we are always welcoming in our group to new collaborators or people who want to explore these questions.
And over to Josh.
Hi, everyone. I'm Josh Kornbluth, and I'm a performer and a filmmaker, and I'm honored to be on this panel, particularly seeing that I'm pretty confident I'm the only person on this panel who flunked all of his science classes in college. And what I actually try to do is act as a conduit between the incredible science that's done at the Memory and Aging Center and elsewhere and the general population. And one of the ways I do this is through a series of 10-minute videos that are available for free online. The series is called Citizen Brain, and I want to communicate recent findings and knowledge about brain science and brain health, but I also have a special interest in connecting brain science and brain health with social justice. And so each of the videos has its own topic, as you can maybe see in this slide, but the first one I did was on the empathy circuit. And it also, among other things, told the amazing story of how the empathy circuit was located through this study of people who have behavioral variant frontotemporal dementia. Another one, Age Without Ageism, which was a particular emotional interest to me as I'm rapidly aging. The loneliness paradox, about loneliness, which besides having scientists, also had two of our stars of the video were C. elegans worms and prairie voles, who both species shared their deep knowledge about what it is like to feel isolated. Then othering and belonging is the most recent one that came out, and it sort of encompasses all of them. It's this idea that in order to have a world that works and that survives and a democracy that flourishes, that we need to include as many people as possible into us and as few people as possible into them. And the one I'm working on now is about certainty versus doubt. Roughly speaking, left brain versus right brain, but not really versus, but how they have to work together. Because if you have too much certainty and you're all the way over in your left brain, you become a zealot and an ideologue. And if you're too much over in your right brain, you're just worried about and doubtful about everything, and you don't do anything and just sit in the corner of your room. So that's been a really fun series for me to do. You can find all of them and me at citizenbrain.org. And I also do one-man shows, and I have a one-man show that I'm doing now on various Zoom platforms. That's also called Citizen Brain, that connects democracy and brain science, and especially the story of how my mom fell in love for the first time in her late 70s with a man who then tragically developed Alzheimer's, and how it's all connected. Thanks.
Thank you. And with that, we're going to pass it over to Virginia, who's going to share her own screen so you can see her nice animations.
Thanks for having me. Can you hear me okay? Can you hear me? No?
We can't hear you, Virginia.
Can you hear me now?
Yeah.
Okay, my microphone is being very temperamental these days, sorry. Okay, thanks for having me. I'm Virginia Sturm. I'm an associate professor at the Memory and Aging Center at UCSF, and I'm going to briefly tell you about our work on emotions in degenerative disease and in healthy aging. So there's lots of ways people think about emotions. This is how we think of them. They're brief phenomena. They have psychological and physiological components associated with them. So you move your face in certain configurations when you feel emotions, and in your body, there are certain changes that happen, such as changes in heart rate and breathing and sweating that accompany different emotional states. And these different emotions are produced by the brain via a distributed system, which is shown here. The details aren't important, but what is important is that we can study this system in a laboratory environment at UCSF, and we can look for how different diseases and disorders affect this system in different ways. So in BVFTD, or behavioral variant frontotemporal dementia, this network that produces emotions is atrophied and has lower connectivity. And in contrast, in Alzheimer's disease, the opposite pattern is found, where this system is hyperconnected. And so this would lead us to different hypotheses about how emotions might be altered in different ways in different forms of dementia. So just a brief overview of some of the examples of things that we found using this approach in behavioral variant frontotemporal dementia are certain emotions are impaired, such as embarrassment, and this relates to atrophy in the cingulate cortex. Disgust is an emotion that is impaired in BVFTD that relates to insula atrophy. And we've also done work showing that even at rest, there's alterations in parasympathetic and sympathetic activity in BVFTD that relate to different patterns of brain function. In Alzheimer's, we've seen the opposite pattern where certain forms of empathy, these very simple forms of what we call affect sharing, so the mirroring and simulation of others' experiences via these autonomic and behavioral systems, are actually enhanced in people with mild cognitive impairment and Alzheimer's disease. And this also relates to different patterns of brain atrophy. And finally, sometimes we look at how emotions are functioning in our everyday lives in healthy people. So not only do emotions change with dementia, but we can also modify our emotions to improve our aging trajectories. So this is a study that we recently published on the emotion of awe. And awe is a positive emotion that you feel in the presence of vast things that you don't immediately understand. So these are some pictures that our participants sent us of things that they noticed in the world around them when they took weekly outdoor walks for 15 minutes for 8 weeks. And they tried to experience more awe in their lives by attending to the details of the world around them. And by doing so, they experienced a number of emotional benefits, including most notably for us, this increase over time in what was called pro-social positive emotions. These are other-oriented emotions such as compassion and admiration and gratitude that increase on a day-to-day basis in our people who took awe walks, suggesting that even in older adults, a simple shift of attention to pay attention to the wonders of the world in front of our noses that we sometimes fail to appreciate can have enduring benefits for our mental health. Thank you.
Great, thank you. And so I know that was brief and a lot of different research, but we really just wanted to highlight there's so many different ways we look at brain health, whether that's intersection with law or genetics or imaging biomarkers or pathology. And it really is extremely integrated here at the Memory and Aging Center. I couldn't recommend working here more. And so while we cover a lot of topics, it really is all embedded all in one place. And we work really hard to make sure any project people are interested in doing, we can do. And with that, I think we have about 10 minutes for questions.
Thank you so much, everybody. First of all, congratulations for the great work. It's very impressive. I was taking notes with all the questions. I think that's probably going to give the opportunity to the audience to ask questions, but it's very rich, and congratulations for all the work that you're doing. Tamara has a question. Tamara, you can ask the question now. Or no? Yeah, okay. So yeah, she disconnected. Okay, any questions from the audience? You can even raise your hand or just type it in the QA. And in the meantime, I can ask one of my questions. So I wanted to ask something that I didn't hear, or maybe I missed it, but any research that you are doing with sleep, especially when you are thinking about resilience and so far. So I don't know if anybody can comment on that.
Kaitlin, go ahead.
Yeah, either one. So absolutely, our center is also very interested in sleep. I think when we think about the biological pathways of resilience, sleep is probably its whole own pathway. And so that's why we have sleep researchers dedicated to this. The work is led by Christine Walsh and Tom Nyland, and they have in lab as well as at home different techniques to profile sleep, including polysomnograph, a couple of sleep intervention trials as well. So this is also definitely a factor we're very interested in. So if folks are interested in quantifying sleep, I think there's a very rich data set available here too.
Yeah, and I wanted to add also some of the work that Leah Grinberg and her lab is doing, looking at neuropathology and correlating it with sleep during life and trying to understand how degeneration in different brainstem nuclei can explain really quite different sleep disorders, even in the balance between hypersomnolence versus hyperalertness. You know, sleep disorders have been really studied more in the context of aging and AD, but less in other types of dementia, and yet they are you know, in Lewy body, of course, as well, but there are really one of the major issues that patients and care partners identify as quality of life issues and symptoms of neurodegenerative disease. So it's a great question, Yakil.
Yeah, no, thank you. Yeah, anybody has any questions? Otherwise, I can just keep asking questions for the next 8 minutes. Just make sure to raise your hand or type the question if you have any questions over there. So as I mentioned before, so I was very impressed by all the work that you guys are doing. And since you are like a very large group, I was wondering, like, I don't know if you can speak a little bit about how you actually work together, you know, how could you actually make sure that everybody knows what the other ones are doing and like, I don't know, like avoiding overlapping of projects. I don't know, any comments on that?
Yeah, it's a great question. I think the ADRC is one of the major ways that we all come together. So we have an executive committee meeting every week where, you know, leaders of different research programs, all our cores get together and discuss things. We have our weekly MAC Grand Rounds, which the ADRC is very prominently featured in, where we have lectures from people leading different research projects within our center. So that's another way that we can communicate with each other. Of course, we also have external speakers. I think, Yakil, you've been one of our speakers at that back when we did this in person, but so that's another thing. And then I think there's just a lot of connectivity between different programs. So Renaud highlighted, for example, just in PET, how we interact very closely with neuropathology, with biomarkers, with genetics. And so, you know, there's a lot of sort of mutual lab meetings that happen, but it is a challenge as the center gets bigger, you know, to maintain that connectivity. I think, you know, the great thing about this place, though, is that, you know, you really have such a broad degree of expertise in all sorts of aspects of ADRD research, ranging from, you know, basic science, molecular genetics, all the way to, you know, care models and policy. And so it's really just a great joy to be in that intellectual milieu. And of course, like everyone else, we've been, you know, trying to figure out ways to stay connected during the pandemic, because a lot of the natural sort of connectivity that happens just in, you know, meeting together and sharing a physical space is not available to us now. So we've been working very hard to try to maintain those interactions. But of course, we're excited to get back and see each other in real life, hopefully very soon.
Thank you. Renaud or Gil, you have a question in the QA. You want to answer that one?
Yeah, I think it's a really, you know, it's a really cool question. Johnny is asking whether we're talking about the heterogeneity of patients with AD and asking if we use like basically how we approach that question. And I think the point that you were making is really interesting and important. And actually we do it both ways. So we do sometimes look at you know, subtypes of patients that are clinically defined. And we published actually a few papers on this, looking at different syndromes and how they differ or resemble each other in imaging. But we also use some other kind of like bottom up, top down approach to use the data itself to stratify patients into subgroups and maybe discover new subtypes of patients that are not already described clinically. So I think we do it both ways depending on the papers.
Yeah, it's interesting. You know, a lot of people say, you know, and when our papers are reviewed, you know, your cohort is really weird, right? You know, your AD patients are young and they don't have memory problems. They have, you know, visual problems and language problems. And I actually think it's a big strength because I'm not trying to claim that our cohorts are representative of the overall, you know, general sample of AD in the population, but it does really allow us to address these questions about what drives heterogeneity in the disease. Why is it that in some people, you know, language networks or visual networks or even behavioral networks are most susceptible to the disease? So that's been a real fun part, is that we're a little off, you know, at San Francisco, we have to be a little offbeat in what we do. And so being off the, you know, traditional late onset AD track has actually, I think, been very rewarding. And I think these patients really have some fundamental principles to teach us about mechanisms of disease that will translate into therapeutic approaches. So embrace the weird, as we say in the Bay Area.
Thank you. I was going to ask you, Gil, because you was mentioned the ideas project, but I don't know if you can tell a little bit more about the ideas and what that is.
Yeah, I'll be very brief. So, you know, amyloid PET scans are approved for clinical use in the US and many other countries, but in the US, insurance doesn't pay for it. Medicare has been trying to decide whether they should pay for it by asking whether doing PET scans in working up MCI or dementia can actually help improve patient care and outcomes. And so that's the basic idea behind ideas. Sorry for the pun. But it's really a national program, very large numbers of patients. And in our next round, which is called New Ideas, we are actually specifically looking at diverse populations, African Americans, Latinx populations across the country to try to understand whether there are specific communities that might even benefit more from amyloid PET imaging and how this might affect outcomes in different ways, depending on different social determinants of health. And actually in New Ideas, we're linking this to a biorepository to really try to determine if some of the polygenic risk scores that Jenn is working on, or blood biomarkers for A-beta, tau, et cetera, how these perform in a real clinical care population and a diverse population. So that study is just now getting started. And Yakil, we should really talk offline about how we can promote the study together. I think it's, you know, some of the organizations that you're working in through the centers could really help with this study as well.
Sure, happy to talk more about that, Gil, for sure. And the last question that I'm going to ask and see if anybody has any other questions. You also mentioned like the brain bank, right? And I was wondering, since you are so interested in diversity, how diverse is the brain bank? And if you have had any issues getting brain donations from diverse communities?
If Serggio, or is Serggio still on, or Boon Lead, do you want to address that? Or I can address it if you're not prepared to.
Well, I can add some things. I think we are around 2 to 4% being a non-white participants at the moment in our brain bank. But I think this is a continuous progress where we are working to promote with community partners that are we have worked with community partners that are particularly focusing on palliative care in dementia population. So that would bring up the conversation about brain donation. And there's also always a time lag in between them consenting now to when they would donate their brain. So we are very optimistic that things would change gradually as we incorporate more and more community partners to work with us on promoting brain donation in the diverse population.
And one thing I'll add to that, that's also something that people research that we didn't talk about, is why people might decline brain donation. And then working with our PATH group and then our outreach group, facilitating different partnerships that might be more interested in helping break down those barriers. I think that's all I want to say there. And then another aspect of diversity is we have a very diverse brain bank in terms of types of clinical syndromes beyond race, ethnicity.
Perfect, thank you. And with that, I would like to again thank you everybody for joining us. Thank you for everybody that came to meet the UCSF. It was really a great pleasure to host you, and have a good day. Thank you, everybody.
Thank you, bye.
Thank you so much.